Well, thank you for the opportunity to speak here. And I would like to say that we will be shifting gears a little bit because this technique I'm going to talk about has not made its way into clinical practice yet. So what we will be discussing is mostly research studies, and hopefully by the end of the talk we might have some ideas on where DTI connectonomics may be really useful. And one of the areas that we are working to understand is whether it would be really helpful in understanding early Alzheimer's-related changes in the preclinical stages of Alzheimer's disease. And by knowing how it relates to tau and amyloid imaging, I think we can make a good case. Now, I would like to start with an outline of what we will be covering here. First of all, we will be talking about some of the basic principles. I'm sure many of you are familiar with them, but it would be good to kind of go over them before we start discussing more sophisticated analysis with these metrics. And pattern of diffusion tensor imaging changes in the Alzheimer's disease spectrum, and what I mean by that is the preclinical stage and then the MCI stage and Alzheimer's dementia stage. And we will move on to the relationships to tau deposition and A-beta deposition and DTI connectonomic signatures. And finally, we will talk about some of the frontiers of this technique, such as the whole brain structural connectonomics and how that might help us understand the brain connections better. Now, what are we really measuring with DTI? First of all, we need to understand what we're sensitive to. It's this random motion of water molecules that occur in the tissue that this DTI technique is sensitive to. And one of the metrics that we are interested in is the mean diffusivity that depends on the integrity of the structural barriers that restrict the motion of these water molecules. And these are membranes of neurons, myelin, intracellular organelles, and macromolecules. And this is just a simple cartoon of how water might be diffusing when there's intact brain tissue with lots of restriction due to cell membranes, and even in the intracellular space there is some diffusion, but in an intact cell this diffusion is relatively restricted. Whereas when we lose the neurons, axons, and myelin, the space of this diffusion, of course, increases, and therefore the water diffusivity, as we measure with mean diffusivity on DTI scans, also goes up. Now, another metric which is particularly useful in white matter is the fractional anisotropy, or I will be referring to it as FA, and this is a measurement of the directionality of water diffusivity. As you know, water diffuses in a certain direction in the white matter across the trajectories, and it depends on the integrity of axons and myelin that determine the direction of the diffusion of water molecules. Now, in an intact white matter where the axons and myelin are healthy, this diffusion occurs in a so this diffusion occurs across and it resembles more like an ellipsoid having one dominant direction. In this case, the FA value is somewhat close to 1, quite high, whereas when we lose axons and myelin, this diffusion is not as directional as the intact white matter because there is now more space for water to diffuse in the perpendicular direction to the axons. And because of that, the diffusivity tensor now resembles more like a sphere than an ellipsoid, which would decrease the directionality measurement we have, the FA, down to maybe half of what we see in the intact white matter. Now, when we look at Alzheimer's disease, comparing a group of Alzheimer's disease patients to cognitively normal controls, in the gray matter, we do see increased diffusivity in this pattern, which resembles very much the pattern that we saw earlier of the structural changes in the brain. And that's not surprising because atrophy and diffusivity go hand in hand. And as you can see, mostly the medial and lateral temporal lobes are involved, as well as the parietal lobes. Now, when we look at the white matter in these patients, what we see for the fractional anisotropy decreases is the underlying white matter that we saw earlier has decreased FA values in some of those same regions of gray matter that's affected. So the pattern of FA decreases in the white matter is very similar to the pattern of gray matter mean diffusivity increases topographically. Now, do these increases in mean diffusivity occur at an earlier stage of Alzheimer's disease? And to investigate that here, a group of non-demented elderly individuals were looked at, and the memory scores were correlated with the gray matter diffusivity in the whole brain. And as you can see, it was the hippocampus that correlated best with the memory scores and found to be significantly affected in these individuals in terms of memory. So Dti changes in the gray matter appears to be correlating well with what's happening earlier in the non-demented older individuals. And this is the map of the language function relating to mean diffusivity changes, and it happens to be more in the left temporal lobe, as we would expect, and correlating mostly with the anterior temporal and inferior temporal gyri. Now, we also correlated these white matter FA findings with other cognitive domains, and here is one of the tracts that's really an important connectivity tract for the temporal lobe and memory function and language function. As you can see, with decreasing memory function, we see lower FA values as well as language in the temporal lobe tract. In the anterior cingulum, which is in the frontal lobe, again, there is a correlation with memory, but also with attention and executive function, as one might expect. In the posterior cingulum, interestingly, the correlations occurred with all cognitive domains that we studied, such as memory, language, attention, and visuospatial function. And that's really not surprising because posterior cingulate gyrus has been implicated for the connectivity hub for all cognitive brain networks in fMRI studies, resting state fMRI studies by some of the earlier publications by Buckner and Gracious. Now, as we have seen, decreased FA may have an important role for early changes in Alzheimer's-related cognitive domains, and also it might be abnormal in preclinical stage of Alzheimer's disease. And here what was done is there's a group of preclinical Alzheimer's disease patients who have amyloid positivity, and also they are positive on neurodegeneration biomarkers such as FDG PET or structural MRI. And in this group, we see that there is a reduction in fractional anisotropy in the fornix compared to a biomarker negative cognitively normal group. When we look at mild cognitive impairment cases, they still have this fornix abnormality. In addition, they show more white matter FA reductions, and these are people with neurodegeneration positive biomarkers. But if they have, in addition, amyloid positivity, these abnormalities grow even further, almost covering the entire white matter, very similar to the pattern we see in Alzheimer's disease patients. Now, for this part of my talk, the summary is that the integrity of white matter is disrupted as early as the preclinical stages. And these DTI alterations are initially localized to the limbic connections and tend to spread to the temporal and parietal white matter areas as the clinical symptoms progress. Anatomically, involvement of these tracts follow this topographic progression of cortical degeneration, atrophy, and metabolic disruption in AD, starting from the medial temporal lobes and spreading to other temporal and parietal areas. Now, I would like to focus on these limbic connections that we found to associate with preclinical Alzheimer's disease. And in this nice atlas that was developed by Mori and Agarwal, which consisted of tractography measurements that are overlaid on a template, and then an atlas was created from that template to be used in detecting the diffusion changes in individual subjects. So this atlas includes the fornix and the cingulum tract, as shown here. It also includes the entorhinal cortex white matter, which is not shown here, but the entorhinal cortex white matter includes the perforant pathway. So kind of reviewing what those tracts might be connecting to, we have the entorhinal cortex connecting to the CA1 subiculum subfields of the hippocampus through the perforant pathway, and fornix originating from the hippocampus and connecting to other limbic structures such as the mammillary bodies and septal nuclei. The mammillary bodies projecting back to the cingulate cortex through the anterior nucleus of thalamus, and then the loop is closed with posterior cingulate cortex connecting to back to the entorhinal cortex through the cingulum tract. So with the atlas-based measurements, we are able to measure the perforant pathway diffusion imaging connectivity. We can measure the cingulum connectivity and also the fornix. Now we will be looking at these tracts more closely and their relationship to tau imaging in the next few slides. So why these tracts might be important in the course of Alzheimer's disease is something we can understand this figure, which was modified from the Braak and Braak neurofibrillary tangle staging of Alzheimer's disease. As you can see, the medial temporal lobe is involved severely and earliest, which then spreads into other paralimbic cortices and then to neocortex, association neocortices, and the primary sensory motor and visual cortex is affected at the end of the disease. And as you can see, the tracts that connect these medial temporal lobe areas are the tracts that are mostly involved with our DTI measurements, which are the fornix and the cingulum here, as well as the inferior longitudinal fasciculus. Now, we have investigated whether abnormalities in these tracts really correlate with the Braak neurofibrillary tangle staging of Alzheimer's disease. Now with the tau imaging, which will be mentioned later, it might be possible to do these studies in vivo. But at this time, we had this group of patients who had ante-mortem diffusion tensor imaging and had post-mortem Alzheimer's disease diagnosis at autopsy. And we looked at their mean diffusivity and FA values in those tracts that are particularly involved in Alzheimer's disease. And when we correlate those with Braak neurofibrillary tangle stage, we see that those limbic tracts are particularly involved with the staging and amount of tau pathology in the brain. So what are those tracts? The cingulum here, precuneus white matter, the splenium of the corpus callosum, entorhinal cortex, white matter, the perforant pathway, and the inferior longitudinal fasciculus. Now, you see the corpus callosum kind of cut right there because the atlas had different portions of the corpus callosum in it, and only the splenium correlated with Braak staging. Now, looking closely at the data, we see that the perforant pathway and the 6 Braak stages here, fractional anisotropy and mean diffusivity, the fractional anisotropy is decreasing with increasing Braak stage, and the mean diffusivity is increasing with increasing Braak stage, as you can see here. Similarly, we see that pattern in the cingulum tract and also in the precuneus white matter. So we will be looking at these fractional anisotropy findings a little more closely and try to understand when these changes occur in these tracts. You can see that there is Braak stage 1 to 2 here and 3 to 4 in the entorhinal cortex fractional anisotropy. We don't really see much difference in Braak stage 1 to 3, whereas when we get to Braak stage 3 to 6, we start to see decreases, and these stages don't seem to be too different from each other with low FA values. So this is very much expected because at Braak stage 2, the tau pathology starts going out of the entorhinal cortex into the hippocampus. And at this stage, what we see is the perforant pathway is getting impacted. So the connections that are in between the tau pathology that's spreading from the entorhinal cortex to hippocampus are being affected. When we look at the cingulum tract, we see that at stage 1 to 3, there's not much difference. And then at stage 4 to 6, we see a significant drop there. And that's also expected because at Braak stage 3, the pathology starts to get out of the hippocampus into the paralimbic cortex. And that's where the cingulum that carries those fibers to the paralimbic cortex, the cingulate gyrus, appears to be affected. Interestingly, in the precuneus, we did not see such a divide between Braak stages. It seems to be a very linear correlation in the precuneus, with highest R value was observed there. So this will require other functional MRI studies, resting state fMRI studies, to understand why precuneus is involved throughout the Braak neurofibrillary tangle stage, and why is the structural change happening starting from the earliest and going on to latest stages. And perhaps fMRI studies looking at functional connectivity in this region may explain some of these findings. Now, in summary, the topographic pattern of gray matter atrophy in Alzheimer's disease, as you know, follows the Braak neurofibrillary staging of Alzheimer's disease pathology. And it seems like a similar pattern exists for the white matter diffusivity changes involving the limbic projection and the medial temporal and parietal white matter regions similarly. And therefore, white matter and gray matter degeneration in Alzheimer's disease appear to be topographically concordant and likely linked to each other. Now, how about A-beta deposition? What is the relationship between these DTI changes and A-beta deposition in Alzheimer's disease? In several studies in the literature, we see that DTI abnormalities are associated with beta amyloid load on PET scans in non-demented individuals and even in clinically normal adults with a family history of Alzheimer's disease. In the Alzheimer's disease neuroimaging cohort, in clinically normal MCI, higher beta amyloid deposition on PET was associated with lower FA values in the fornix and splenium, and the cognition was only affected when there is an increase in A beta load together with a decrease in white matter FA. And that kind of shows us that white matter FA changes are critical when they occur with beta amyloid increases. Now, to investigate that, we looked at our Mayo Clinic study of aging cohort, which is a large cohort of 700 people with ages 70 to 90. And here we have a group of cognitively normal individuals. About 1/4 of them also had MCI. And in this, for these groups, we divided our sample into those that are amyloid positive and negative, and those that have neurodegeneration positivity and negativity. And what I mean by neurodegeneration, again, is atrophy in the gray matter that is similar to an Alzheimer's pattern, and also FDG PET changes, again resembling the Alzheimer's pattern. So we had this biomarker negative group in the cognitively normal controls, which was used as the control group for comparisons. And when we compared each one of these groups in the MCI and cognitively normal individuals to this control group, what we found was that the amyloid positive groups that do not have neurodegeneration and the biomarker negative MCI group was not different from our control group in terms of FA changes on DTI images. Whereas in the amyloid negative or positive but neurodegeneration positive groups, so when there's gray matter neurodegeneration, we also see white matter changes. It doesn't matter whether they're amyloid positive or negative. Now, in looking at this neurodegeneration positive group and trying to understand what the pattern was, what pattern of changes occurred in these people, here we have the neurodegeneration positive but amyloid negative group compared to the biomarker negative controls. As you can see, the regions that are affected is the fornix, the posterior parietal white matter, and the splenium of the corpus callosum. When they're amyloid positive, these changes also occur in the same regions, but it's a little more extensive. The pattern is not changing, though. Now, in the mild cognitive impairment, we have the neurodegeneration positive and amyloid negative group, still very similar to this pattern we see in cognitively normal individuals, but more extensive, and even more extensive involvement in the MCI patients who are both neurodegeneration positive and amyloid positive. So with additional biomarker positivity, we see the white matter involvement growing. And when we look at some of those plots, you can see in the fornix that the decrease in FA is very significant in the neurodegeneration positive, but not neurodegeneration negative groups, similarly in the MCI. But presence of amyloid positivity does not seem to be impacting this. So in the neurodegeneration negative normals, you can see amyloid positivity is not causing a decrease in fractional anisotropy when there's no gray matter neurodegeneration. So in this investigation of fornix, we can say that high amyloid load by itself did not have an effect on microstructural integrity of white matter in the absence of gray matter injury. So the effects of beta amyloid on white matter appear to be linked to gray matter injury, and those who have both gray matter neurodegeneration and amyloid biomarker positivity tend to have more widespread white matter FA decreases than those who just have neurodegeneration. So it appears that these neurodegenerative changes go hand in hand with white matter degenerative changes, and presence of amyloid even increases this involvement. Now, finally, I would like to talk about some of the new research coming out of the whole brain connectome, and this is considered to be frontier of DTI. We see these really pretty images in social media and other journal covers, and they look really nice, and they kind of tell us what is going to be happening in the future. But how can we use this information to understand the brain connectome better? Now, the structural connectonomics, which is an emerging area, is about mapping of connections within neural networks. And it's done by application of this graph theoretical analysis to DTI tractography and mapping structural brain networks. I won't go into the technical details of this, which was discussed in a session that happened previously on brain connectonomics. But simply put, by placement of nodes in the cortex using tractography streamlines as edges, as those that connect those nodes, the goal of this approach is to perform quantitative analysis on the organizational structure of the connectome. And there has been a few studies in Alzheimer's disease, and I wanted to show this, which looked at beta amyloid load relationship with the structural connectome of Alzheimer's disease. And what was found that the beta amyloid load on PET was associated with disruption in the structural connectome, particularly in frontal, temporal, and parietal regions, and those are, of course, the regions that are most affected in Alzheimer's disease. And they found that reduced network strength, which is the connection between those nodes, and the local efficiency, which is the strength of those connections, how close they are, and then the weighted clustering coefficient, which is those nodes that cluster together, how well do they cluster, is found to be lower in Alzheimer's disease patients. Now, finally, I have a few take-home messages. And first, disruption in structural connectivity in the white matter is associated with the gray matter degeneration and Braak neurofibrillary tangle stage in Alzheimer's disease. We discussed that limbic tracts, particularly those that connect to the medial temporal lobe, are affected during the preclinical stage and involve rest of the white matter as the disease progresses. Whether DTI can detect neurodegenerative changes associated with Alzheimer's pathology earlier than gray matter atrophy on MRI or cortical hypometabolism on PET requires longitudinal studies on large cohorts that transition from preclinical to clinical Alzheimer's disease. And connectonomics of the brain may open new frontiers in understanding the cognitive decline, brain reserve, and progression of cortical pathology through the structural connectivity networks in Alzheimer's disease. So thank you for your attention, and I think, I hope I was on time.
Wonderful. Thank you so much.